Thanks for coming. Thank you. Yeah, so this morning we're going to talk about something that basically revolutionized how we work in weeds. It was quite a long process. It happened over like the past 10 years, but as you can see from the slides, by no means have we reached peak, genomic resources yet. In fact, what we noticed over the last few years is that the pace is actually accelerating. So, it's Philippa, Andy, I don't know if Cristobal will say anything, James and Catherine, and I'm Nikolai. We'll try and give you like a brief overview of these resources. We don't have time to go into all the details now, but we provide sort of like links and some resources for you, so you can go, explore more on your own. So the way this morning is designed is we'll do some theory, and we have a little coffee break to sort of like refresh yourself, and then we set aside an hour for some practical exercise, because we find that with this resource, if you just talk about it, it's no good. You have to sort of get stuck in yourself. And yeah, we're covering sort of like broadly genomic resources, pangenome, expression data, mining gene networks, and variation SNP data. And so we start with an update on the current state of wheat genomics. And as I say, things have, like 10 years ago, there were very few genomic resources. And there's a reason for this, because the challenge of the wheat genome is severalfold. So Arabidopsis thaliana is a modern plant. It was sequenced in the year 2000. Has sparked a huge revolution in gene discovery and whatnot, and has a really nice compact genome of 125 megabases. Rice was assembled a little bit later, and that's because it's slightly bigger, so the challenge is slightly bigger, so it's around 400 megabases. Human genome is around 3 gigabases, so it's quite extensive, also around the millennium that we have the assembly. But then you look at wheat, it's a 15-16 gigabase size genome, so it's 5 times as large as a human genome, and that was, that took a lot of effort to sort of get sequenced. In addition to that, we have some other compounding factors. One being that the hexaploid wheat genome is, well, a hexaploid, so it came about so it's an allopolyploid, meaning that 3 diploid progenitor species were combined in a single species. So you actually have 3 separate genomes in there, all with their own genes. So usually it means you have 3 copies of every gene. But then the majority of these 16 gigabases is actually repetitive, and that tends to be the biggest hurdle. That about 85% of the genome is repetitive, retrotransposons, DNA transposons, and it was just really difficult to make sense of it, assemble this genome, because you have a gene, maybe three genes, and then you hit a long stretch of retrotransposons again. And how do you put this all together? I'm going to touch briefly on this. And because the genome is so complex, and we're going to talk about these different genes, we need to know how they are related to each other. I'm just going to do a brief recap on gene homology. So I use the term homeologs, which I love. It's ONEs. It's really nice to pronounce. So we talk about homologs. These are genes from similar chromosomes brought together through interspecies hybridization or allopolyploidization. So allopolyploid, that's the wheat genome. So your 3 genes, they are homologs. So if you look at the wheat genome, we have our 3 subgenomes, A, B. You can expect that any genome, A genome has copies on the B and D genome as well, and these are homologs. We're also going to talk about orthologs. So these are genes from different species that were from a common ancestor. So, for example, if we compare rice and wheat, they have a common ancestor, but they're 35, 40 million years ago. We have our homologs in the wheat genome, and then if we try to find the corresponding gene in the rice genome, that would be the ortholog. So this gene is the ortholog of those 2 genes, and vice versa. Right? And lastly, with paralogs, these are genes that result from gene duplication within a species. So, for example, in wheat, so again, these are 3 homologs that exist in the wheat genome. And, this gene on the H genome actually duplicated within wheat itself. So it's not present in rye, not in barley, not in rice. This happened only in the Triticum moratum, so in the progenitor. It was passed into wheat. So that's what we call a paralog. All right. With this out of the way, let's get into it. So I'm going to talk about 3 generations of wheat genomes, because that's how many we had in the past 10 years. And, gigantic efforts by both academic partners, industry partners, and yeah, as I said, I'm going to touch briefly on all of this. So around in the year 2014, 8 years ago, doesn't seem so far ago, but in terms of genomic resources, it's like the Stone Ages almost. We had the very first assembled genome of wheat, and this was called the Chine Spring Surface Sequence, CSS. We had roughly 1 million scaffolds and context, so the wheat genome did not exist in like chromosomes or anything, but you had like 1 million scaffolds, and they tend to be fairly small, on average 7 kb. These only covered about 10 gigabases, so around 2/3 of the wheat genome. So not everything was actually present. And because you have these main scaffolds, and I said retrotransposons, repetitive elements are difficult. So because of this, these scaffolds only contains mostly like genic regions. But you couldn't put them in like prop order. We had like a crude order, but not a great one. But they were good enough to actually design or annotate the scaffolds. So we have gene models for them. And you might still find in the literature some of these gene models, hence why we present it here. So just to explain a little bit the idea behind those gene models and what information you can get from them. So this is just this name looks like this. You have this trace underscore 1BL. And underscore and the number. So trace is for species, Stevum. 1BL in this case denotes the chromosome arm the gene model is on. So there's fantastic back in the time. You could immediately tell, oh, this gene is in this location. Previously, you worked with ESTs or something, and you had to figure out where a gene is. And lastly, you have sort of a unique ID. This is no position information. So all you know is that this gene is in chromosome 1BL, but where it is. Little warning sign. So while this is the standard for this genome assembly, chromosome 3B was assembled separately in a separate project, and a separate project made their own separate gene models. So while this for all the chromosomes except chromosome 3B, this is how the gene models look like for that assembly, the CSS. But all the 3B gene models look like this: trace 3B, and then a really long number. And you see throughout this presentation. Unfortunately, this is kind of a trend that you have sort of one set of gene models and then someone else comes along and puts up another set of gene models. Right, okay. Around 3 years after the CSS, we had another evolution in the genome. So in 2017, Renato Clavijo from TIGAC, now EI, led an initiative to make a new assembly of the wheat genome. Slightly fewer contexts, so around 700,000. The big improvement was that these context scaffolds were a lot bigger. So on average, like 90 kilobases, so 10 times larger. So suddenly you could have maybe several genes on contact rather than like one, just a partial gene. The order of these scaffolds, because they were longer, you could more easily order them. So you had sort of like rough, large bins that you could place the scaffolds in. And of course, it comes with its own genome assembly. And it looks like a mouthful, so let's go through it slowly. So again, we start with Trial is the species, and then CS42, that's the accession, so that's Chine Spring 42. Again, we have information where this gene is located in the genome, so in this case, chromosome 7BL. We have the assembly version, this is TGFB1, so they only made this one assembly, so this is the only assembly, or the only assembly version you can find. You have another number which denotes which scaffold this gene is on, and then the gene number. So already you have much more information than in the previous set of gene models, and with that information you can actually figure out, okay, if you see this name, you can figure out which scaffold in the genome it's from, because the scaffold names are basically like the assembly version scaffold, the scaffold number, and the chromosome number, it's on. So with this information, you can always say like, ah, I know this gene is on this scaffold. Fantastic. And then the big revolution came very, very shortly after, actually, with the RefSeq genome assembly. So suddenly we are moving away from scaffolds, and we are actually going to pseudo chromosomes, 21 pseudo chromosomes, one for each chromosome, and then there were some sequences that could not be assigned to chromosomes, so they were put in a 22nd chromosome, which is chromosome UN, unassigned. And almost the entire, or a large proportion of the wheat genome is assembled now, 46 gigabases. It's almost everything. Because we have these large pseudomolecules, the way they were assembled, we had information, or you have to have information about the position of each of the scaffolds to put them into a pseudochromosome. So for this assembly, for the very first time, we have like a true, in quotation marks, physical order. And then, of course, really good gene models were called on this assembly, because the assembly quality. So the quality of the gene models always scales with the quality of the assembly. The gene models from the CSS assembly were crude, most often wrong, partial genes, and that was down because the assembly was so poor. And as the genome assembly improves, the quality of the gene models improves significantly. And now we actually have really good gene models. And just talk you through how they look like. Again, it's a very, very similar structure. So the name, you start with the trace, which is the species, CS is the accession, Chan spring. You then know just the chromosome, not chromosome arm, so it's not 2BS, 2BL, but just 2B, so you know it's somewhere there. Followed by assembly version, and then the gene number. And essentially, the gene number tells you roughly where the gene is. So this gene here, 100, is the first gene on chromosome 2B that's annotated. So they tend to go in steps of 100. The next gene would be 200, 300, 400, and so on and so forth. For the first time in this assembly, what we also had was high and low confidence gene models. So high confidence gene models are gene models there where there was a lot of evidence from other species, from RNA-seq data, and some other sources of evidence that this gene is real, that there's transcript being made from some genomic sequence, and this is a gene model. But there's also another category which are the low confidence genes, and they have, in terms of the gene name, they have very similar structure. So everything up to gene number is the same, and the only difference is that at the end they have these two letters LC, low confidence. So if you see a gene with LC, you know it's a low confidence gene. That doesn't mean the gene is not correct. This category could include like orphan genes that are not present in other species, so you wouldn't have as much evidence as for like the regular high confidence genes. But also a large proportion of these are actually can also be repetitive in nature, or the pseudogenes, or something else. Right. So word of warning. So if you look at one of these genes and you want to find out, you want to figure out what are the homologs. Homologs are not simply the homologs don't have simply just the same gene number as your gene. No. This gene number simply denotes the position on the chromosome. So this is the first genome to B, this is the first genome to A, this is the first genome to D. That doesn't mean that they are homologous, because there can be rearrangements, whatever, between the genomes, gene implications. So suddenly, the first gene on to B might become the second gene. Okay, something else inserted in front of it. So don't try to find a homolog just by looking at the gene number. It will not work. The other thing to be mindful of is, so you have these two sets of gene models, the high confidence and low confidence. And these are not sort of integrated with one another. So essentially, you can have almost the exact same name in gene models. So here you have TRES CS4B, 01G, 000500, and you have the same LC. This does not mean that these genes are in any way related. They are completely separate genes. If you look at the genomic context, this gene here is the 5th gene on chromosome 4B, and it's preceded by the 4th gene, 0400, followed by the 6th gene, 0600, and then the very first local is present on that chromosome, and that's being called, TRES 100. It's the first on that, let's call it this, meaning that this gene here is the 5th low confidence gene on chromosome 4b. So this gene is roughly 8 genes over from there. So it's not related in any way. It's not a duplicate, not a paralog, nothing. It's completely separate. Okay. Right. Shortly after the new RefSeq assembly, we had an update on the annotation. So this basically a few people come together, re-annotating a few of the pet genes and releasing a new annotation, which is called RefSeq v1.1. In the majority of cases, you can simply convert the gene name from the one version to the other by changing the assembly number from 01 to 02. So you can think of like first assembly 1, or first annotation 01, second annotation 02. Most genes are like this. As I say, there are around like 2,000 genes that were annotated by hand, and these will not simply scale like this. So just double check to be sure, but rough rule of thumb, just change that number and you're good. Okay. I said we had 3 generations of wheat genomes over the last years. It's not quite true. So we actually have 3.5 now, because what happens is we had an update to the RefSeq v1.0 assembly. So there's new assembly out now, RefSeq 2.1. And it's very similar. So the difference is the devil's in the details. So it is a slightly more complete assembly. So there's roughly 168 megabases extra in the genome. It's not much. The biggest change is that for this assembly, they used long read data, optical maps to improve the order and the way the chromosomes are put together. So the biggest improvement is sort of the order. So before the order was true, and I was like even truer. So you know, what does this mean? In the previous assembly, the 1.0 assembly might have falsely, you know, put certain scaffolds in a certain order. With this assembly, they swing around, they correct these mistakes. So you might find that certain parts of the chromosome are now sort of in the complete opposite order than they were in the previous assembly. And that has a knock-on effect for the annotation. So they also made a new annotation for this assembly. So to conclude, so very similar to the previous annotation. The big difference is the assembly version is now 03, the 3rd assembly. And they have an extra number, an extra digit in the gene number. And that's not just because they want to be different. It's because they actually integrated the high and low confidence genes into a single annotation. That means that you can now, that the gene number now tells you exactly the position of the gene on the new assembly, regardless whether it's a high confidence or low confidence gene. For example, here, this is chromosome 1A, the 4th gene. The next gene is a low confidence gene, and it has the 0500, and we have a high confidence gene again, the low confidence gene, and they fit into this. They're completely interleaved. So you have, so you can tell exactly again which what the order of the genes are, both for high confidence and low confidence gene models. All right. So just comparing the 3. So we have the version 1, 01, the version 1.1, 02. So don't get confused by these things. And the 3rd iteration, 2.1, which is 0.3. And again, beware. You cannot, because the high and low confidence gene modes were interleaved, because a lot of the genome, or a certain percentage of the genome was sort of rearranged, the gene orders are no longer comparable. So you can't just simply slap on the 03 and expect this to be the equivalent gene model. You have to find it. So in this case, so what used to be the first gene model of chromosome 2B is actually now the 108th, because the first couple of megabases of chromosome 2B was actually corrected in the new assembly. So it was all inverted. So all the genes are still there. It's just gene order is exactly the opposite to what it was before for that stretch. All right, so just recap briefly. So history of the wheat genome is complex. with 5 different set of gene annotations already for the hexaploid. If you use any genome assemblies, obviously the RefSeq assemblies are sort of the gold standard. Don't use the CSS, don't use the TGAG. You know, everything will be much easier if you use the pseudochromosome assemblies, right? And then for the RefSeq, we have 3 sets of gene models at the moment. Most systems still use the version 1.1, so with the 0.2 in there. You've had still a lot of, or there's still a lot of resources, and we'll show you some today that still use the original annotation, the version 1.0, so 0.1 gene. And also a lot of publications have come out. So you read them and you see the gene models with the 0 G. You know now what this means and how to convert it. And then the newest annotation, the version 2.1, some websites have already adopted it. It's definitely coming. You start seeing people working with it. So that's yeah, that's going to bring about a huge level of change for all of us. All right. So that was the very first region that was assembled. But immediately, you know, straight after you have please ask questions whenever you feel like it.
So, you know, sometimes I read really old paper and they have like TGAG model or CSS. I convert them to a model of RefSeq. So
the TGAG gene models are still within the ensemble plants database. So if you activate your to display alternative models, they still come up with the CSS gene models. There's no really easy way to get it. So you'd have to sort of do a blast and just figure out, okay, this corresponds to that. And most of what you find is that 3 or 4 CSS gene models correspond to the same complete gene because they were very fragmented because the assembly was very fragmented. All right. So we had our first assembly, but right away people turned around, oh, that's not good enough. We need more. So we had a 10+ genome project was initiated. The idea behind it was to create what's called a pan-genome. So essentially, a single reference is not fully representative of all the genes, of all the variations you find within the species, obviously, right? I mean, if you sequence a genome of one of you, it's not going to be represented for everyone, right? Makes sense. So by sequencing many different cultivars, we establish several facts. We establish sort of a core set of genes that's kind of core to species. And we find sort of unique and dispensable bits gene sets. So these are, these might be things that fall on like copy number variation. So certain cultivars having more copies of some, but actually just to live, just for the plant to sort of like survive, it only needs one copy maybe. And also things like presence absence variation. So even in diploids, people notice sometimes you cross 2 accessions together and they are not fertile because they have different presence absence variation. And when you combine them, you actually combine, you create sort of like a lethal combination of either gene presence or absence. And the same can happen in a polyploid, obviously, which is much more plastic in the genome. And by sequencing more lines, we get as I say, we get like a complete atlas of all the genes that have ever existed, as far as we know, in the species. So it's really worth for that level. And so the project set out to sequence initially like 10 other genomes, but it kind of gained momentum and other things were added. So in total, it was 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 hexaploid red meat genomes were sort of taken forward and assembled. 1 genome of spelt, just from spelter, 2 durum accessions, and an accession of wild emma wheat, so one of the primitive species of wheat. And then a subset of these lines were assembled using the same method as the Chinese spring genome assembly. It's called the de novo magic. So for these lines, you have pseudochromosomes. So you have the same sort of quality of assembly as with the RefSeq assembly. And then a subset of these lines was assembled sort of the same as the TGAC D1 assembly from Channel Spring. So that's a lot cheaper to do, and that means these assemblies do not exist as pseudo chromosomes, but as scaffolds. They're still valuable, still can give information. You can still find homologous genes because the scaffolds are good. The gene models on them are like this good. And I mentioned gene models. So for each of these assemblies, so previously all of the gene models were called TRES CS for Channel Spring. But these have their own gene models now. So when you look at the annotation, Lancer is an Australian line. Gene models we call TRES LAC for Lancer. Maize would be MAC, and so on and so forth. Okay. So for each of these lines, you can have up to 2 different sets of gene models. There's 2 lines. So one set of gene models are gene projections. So essentially, the genes from Channel Spring that were annotated were blasted against those assemblies, and applying certain criteria, kind of said, yes, this gene exists in the genome. Because of SNPs variation, it's not very accurate, so you might miss some genes or overestimate gene content. But for all of these assemblies, this exists, so these gene projections. And they look like this. So it's a trace. This is a gene from Lancer, so AC, your chromosome 3A. These are called 0-1G. So these are gene projections from RESIC with the 0-G and then the gene number. No, I do not want to update. Yeah, good. The other set of gene models you can have are de novo gene models. They're called on the assembly specifically. And you have 2 types. So for the genome assemblies they're sort of RESIC standard, so pseudo chromosomes, you have these PGSB gene models. Again, similar structure as always, trace. Again, this is the Lancer gene 3A, and this is denoted by 0-3G. So there's no 0-2G in the pan genome. So you 0-1G, 0-3G, and then your gene number. And for the assemblies that are scaffold based, it's similar to the Channel Spring TGAC assembly. So you have your species trace, then your the accession, this is cadenza cat, the scaffold number, assembly version, gene number. And again, be careful. So this is the gene model projection. This is the actual gene. These genes are equivalent to another. Notice how the gene number has nothing in common whatsoever. In fact, the novo annotated genes have 2 extra digits at the end, so you couldn't even just convert these that easily. Likewise, if you look at the same different pangenome varieties, again, the gene number says nothing whether they are equivalent to one another. So this is so on the top I have the Channel Spring gene from the 1.1 annotation, so on the chromosome 3A, and then equivalent genes in Lancer, Arena, and you see those numbers are completely different. So you can't simply swap the accession name and expect to find the corresponding gene in that. Yes.
If I have a sample of gene in Jagger and I want to find one in Lancer, I can't just swap the number. So what?
Again, the ensemble plants database has the pangenome cultivars in it, and it has tools, or it has all the corresponding relationships between these genes. So you can simply go there, with your, let's say your gene from Lancer. You click on the pangenome tabs and find the corresponding genes in your other pangenome lines. Simple. Thank you. And yeah, so this 10+ Genome project was a huge international project by Carlos Pozniak from University of Saskatchewan, and of course a lot of industry and other academic partners. All right, so you think, surely kind of ceiling. No. Just in the last 2 years, we had more genome assemblies being released. One being really exciting for us, a chromosome scale assembly of the cultivar Fielder. Fielder is one of these wheat cultivars that's very amenable to transformation. So it's really good having this like full sequence of this, because now if you want to make CRISPR lines, you design your guides, you design them on Fielder sequence, on the lines that have been transformed, rather than designing on one of the other genomes and hoping there's no SNPs between them, or having to sort of like PCR out all your intervals and sort of figure out, are there any SNPs or not any SNPs? So really fantastic. Recently, an important French red wheat cultivar, Renan, was again pseudochromosomes. We have an assembly. What was this? Kari, I think, a South African wheat variety. And another assembly of a UK line, Attraction. So it's like, it just never stops. We've reached peak. No. The pace just accelerates, right? So this was in the last year. So it took several years to get like one genome assembly, and suddenly you have so many. It's like buses, right? You wait ages, no one comes, and suddenly you have they're all coming in. All right, we've touched already now some questions from the audience. Already touched on, all this information. Yes, please. No, I just ask questions. How do you access the data? Where do you get it from? And there's many different places, but we kind of recommend, or most of the stuff we do, we kind of use these two databases. We mentioned on some plants already, and there's also Gradient Solution based database. So on some plants, kind of the start screen. This is the website. The most widely used plants and moss species are kind of a different page, so you can just then if you want to select something special, just drop down box and select whatever you want. For example, you want to look at the pangenome lines. Great, they're listed as triticum sequential cultivars. Follow the link. You have your pangenome lines, Stanley, Norin, Lancer, and so on and so forth. And you can go into the assembly about the assembly, so you can see, for example, the genome notation, PGSB, meaning okay, this is the chromosome assembly. EI, Early Institute, that means it's one of the scaffold assemblies, right? Great to see. And again, so let's say you go. The gene that we start looking is red, its homologs are in blue, and then you have sort of the equivalent genes from the other pangenome. So even from so this is Tetraploid wheat, Julius, blah blah blah. So super easy. So you don't have to do any blasting or so. You take your gene name, you take it there, easy. You can do this one by one gene at a time. Or you can go to the Biomart. So as the name suggests, it's like a shopping center for genomic data. So on the there's this little button, very inconspicuous but very powerful. You can click on it. There's different databases that you can select from, meaning either sort of assemblies, or if you want variation data, you select your species. And then it's a huge database. So if you request all the data, you can't make any sense of it. So you need to apply filters. So we'll tap filters. So you can filter, for example, by chromosome, position, type of SNP you're interested in, to sort of like narrow down your search pattern. And you can ask for very different types of outputs. So you can just get like a list of the genes in the interval. You can get the gene sequences. It could be genomic, coding sequence, cDNA. You can get orthologs from your pangenome lines, from rice, brachypodium, because there's a gene in rice that does something, or what gene is in the wheat. I want to know more. So you can get a lot of data very quickly. And this, for example, an example output. So I wanted to know which genes in pangenome cultivar arena corresponds or what are the corresponding genes between ARENA and the Chinese Spring Genome Assembly. Here are all the genes, all the corresponding genes in Chinese Spring, and run them one by one. You get a nice big table, you get sorted and play around with it and have fun and do great research. Green genes is the blood. They're very quick to update the blast portal, so you find all the latest assemblies I talked about. So we have Attraction, Renown, Carriega. So the assembly just came out very recently. It has a trap database, so if you want to annotate a sequence and see what repetitive elements are there, you can use the trap database. You can do sort of like obviously the easiest thing is the most ensemble, but you have the option. So these are great portals I recommend you use. All right, with that, are there any questions to this part of the?
My name's Catherine, and I'll be talking about how you can find expression data for your gene. So why look at gene expression data? Thanks for your questions. It kind of introduced this. For instance, if we have a wheat ortholog of a gene in rice that we want to know, what does it do? Does it do something similar to the gene in rice? Or for instance, we want to narrow down a list of candidate genes that are in a QTL interval. We can look at gene expression data to do that. So gene expression data reflects the level of messenger RNA in the cells. And as messenger RNA is then translated into protein, which carries out the gene function, we can use mRNA level to start to make hypotheses about what the gene function might be. So the first tool I'll show you for looking at gene expression data is a website called wheatexpression.com or Wheat Expression Browser. And it's explained in this paper. When you go to wheatexpression.com, this is the screen you first see. And this browser has RNA sequencing data from 36 studies and over 1,000 samples covering wheat development in different tissues, environmental stress and disease treatments. So how do we use this? At a bit more detail as to what each study is from the Studies tab, and decide which studies we want to explore. And then put our gene name into the genome box. This resource goes up to RefSeq version 1.1. Study we want. For this, I'll look at the Chinese Spring development and click the search button. The default result you get out looks a bit like this. So this bar graph is the gene expression in TPM, which stands for transcripts per million. So the total TPM of an RNA-seq sample is always adds up to the same total. So that kind of normalizes for in case different RNA-seq samples across different studies have a different total, it should kind of balance that out a bit. And then over here, we have a series of categories, including which study, which tissue, what age the plant was, and whether it's had any stress treatments. So we can then use some of these buttons up here to show and hide the categories we're interested in, and click on the columns to sort according to the category that we're most interested in. So for instance, for this one, I started with Chinese Spring development. I'm going to add a few more studies on there. And you can also sort of select exactly which tissues you want to look at. We can then have a more customized view. So here I've I've kind of organized it by the high level. So we've got sort of spike and then grain, and this target gene is most highly expressed in spikes. As Nikolai explained earlier, most genes in wheat have 3 homologs. So what about the 2 homologs of this gene? Wheat expression can help us with that as well. The little homologs button in the top corner. And it automatically shows our 3 homologs. Obviously, it's good to check if these are the 3 homologs you're expecting. This is a quite usual output to see that the A, B, and D genes have all very similar expression profiles. Another way to look at homologs is this ternary plot feature. This represents kind of the same data as the slide before in a slightly different way. So this triangle representing the A in one corner, B in another corner, the D in the third corner. So for this set of homologs, all the points are roughly in the middle, which means that the expression is similar between the 3 genes. And that's what we expect to see again for most genes in wheat. For instance, if you saw all the points towards the top here, it would indicate that the B homolog was most highly expressed across all tissues, whilst if you had a really big spread of points, it would suggest that different homologs are expressed in different tissues. In the example that maybe we have a QTL interval, maybe we have a lot of genes that we want to compare at once, we can also input multiple genes into this browser. This will give us a heat map. So this is our whole list of genes, again, in our different tissues. And this has done a color scale, so the darker color indicates higher expression. The reason this is log 2 TPM is that just makes it easier to see a wide range of expression values that you get when you're looking at multiple genes. For all of these, it's possible to use the buttons at the top to save the images you see, to save the numbers of the data that went into the specific image, and then to save the kind of raw data that's behind that. Now James is going to talk about another wheat expression tool.
Hello, I'm James, and I'll be talking to you about Wheat EFP browser, which is another expression browser. This is a bit more sort of specific. You're looking at a developmental time course, as well as apoptotic stresses and grain development. So it's more of a pictographic representation of the expression in these. And this uses the RefSeq version 1 gene models and none of the later ones. And we're using these 2 genes here as example outputs through the top. So when you set it up, you get an example like this, and on to start with, right at the top here is the input. So what we're looking at to start with here is the developmental atlas with these genes put it in here. And what you get is this output here, which to start with looks very big, and you think, well, why are there so many roots? Why are there so many leaves shown? But this is your developmental atlas here. So we're starting at the top left, nice and logically, with the seedling stage, progressing through multiple different leaf stages, and then moving into flag leaf and full boot up until grain development. And then multiple roots, within the tillering stage, flag leaf, and so on. So there's flag leaf blade. Able to track the expression of your gene throughout multiple time points, multiple development stages within single tissues as well. And the colours here are related to the scale on the bottom. So you see, starting with the yellow, you've got 0 expression, so absolutely nothing. And moving up to the red, which you get the highest. So from this, you can see with this gene that the highest expression is within the flag leaf stage here and full boot along here as well. So moving on to the second gene. So again, same sort of thing, but you see there's a wider range of expression across most different tissues and developmental time points. And at first glance, you think that are red, and so they'll have about the same expression as the first gene you've seen, but that is a pitfall, and you cannot make direct comparisons to this immediately from the default input. So you look at your scale here, and you see the maximum value of 68. And taking it from the first one, the maximum value about 97. So the deep red with that maximum expression is not the same between the two of them. What you can do to correct this is signal threshold option right at the top. And by altering this for both of them, you're able to see the red being the same for both, and so making a bit more comparisons. Alternatively, what you can do with a single gene is to look at the expression data relative to overall expression levels. So this is the overall expression level of this gene. So 0 here, the reference is the median expression of this gene across all different tissues. So here at about 0, we're seeing its bang average expression. And then when we move into the red, it's a higher level of expression, and blue is a decrease in expression. So this is the second gene again, and you're seeing again you're getting, I can't really see it from this angle, sorry, seeing higher expression around. Yeah, I think I've highlighted it. And not okay. But then alternatively, rather than altering your signal threshold like I did in the earlier slides, you can directly compare two different genes. So this is both of the example genes here. So you have input there and they're shown here. So the top gene, this first gene I showed you, is shown in red, and the bottom and the second gene is shown in blue. So what you have with this is where you see red, orange, and yellow is a red expression. You're seeing a higher expression of the first gene. Alternatively, well, so yes, you see the higher expression again right here. But then blue is showing you higher expression of the second gene, which you're seeing around the grains and also along the roots along the bottom. Again, you can see multiple different time points to make that comparison there. That's all wonderful. You've made this. How are you going to get that out and to do something more useful with? So part of the right at the bottom that I cut out from the pictures is you have these two options here. You have expression, a table of expression values as well as a chart. So if you click on that chart, so you've got your table, which has all the different tissues along the different time points, as well as the expression level, and you're able to save that. Right at the top there. Sorry, I thought I cut it out. And then you'll be able to use that for later things. If you don't like, you make your own charts to see the expression, or you can use the chart that they here. So again, you've got all the different time points in the tissues along the bottom. Expression values here. So you can see where the higher ones are, and it's a bit easier to see than the colours if you're not visual. Okay, so that's and those features are common across a lot of features within here. So we're moving on, as I said. So that was developmental time course. Instead, you've got abiotic stress. So you have your gene and you're interested in what its expression is under different abiotic stresses. So again, changing your data source to abiotic stress, and you can input your gene, and you get this chart out here. So what this is looking at is the expression within the flag leaf after the ear has emerged. And you've got your control here, so there's no of the abiotic stress conditions here. And the expression is shown below it. And then with each of these, you've got the application of one of the abiotic stresses, and you've got three together, combinations of two together, and finally all three at once. And again, you can get out the table of expression values, which is a bit shorter for this because there's not as many points, and also the chart like this, which yes, I can show you here. The highest expression you have is heat and drought, as well as go on, enjoy your conclusions from this. And finally, we'll look at the third data source and the final data source for the EFP browser, and that is looking at gene expression during embryogenesis. So to start with, this looks a bit complicated, so I'll take a moment to talk it through. Here, change data source of wheat embryogenesis, and we're inputting the second gene here. As we saw, that had higher expression during the wheat grain compared to the two genes I've shown you. So what you have here is along the top for both of these, and the nice pictures is the hexaploid wheat. So yes, you have expression within the embryo and the endosperm for this. And then you also, along the bottom, you have these rows which have the tetraploid. You have Triticum monococcum representing the A genome here. So for the B and Tauschi for the D. And as you would have hopefully be remembering from Nikolai's talk earlier, you can see the A up in the gene ID there. And so as you'd expect, you're seeing expression hexaploid with the A genome. Tetraploid also has an A genome, and one representative of the A genome. So with this, you can see that there's a varying gene expression. These time points, and the highest within monococcum, and you can export this data into the table again. So with this time, you have different groups. These are representative of the different sort of rows along here. So group one for this one is the A genome. Then you've got hexaploid, tetraploid, and it moves on to the other two. However, there is no expression there, so it's zeros. And also, then you can have the chart. So again, you see no expression for the B and the D, expression for the other three groups. I thought to wrap it all up with a comparison. So with wheat expression, you look at stress, development, tissue samples, and you have lots and lots of different samples you're able to look at. And with that, you can compare multiple different genes. And out of that, you get heat map and histogram visualizations, and your gene models are in RefSeq version 1.1. And then for EFP, it's only a subset of those samples, but you're able to look at the developmental time course, different stresses and embryogenesis. Compare up to 2 different genes, and you get nice little pictographic visualizations. And then for this, you'll use the older gene models of version 1. And both of them have the data available on the Sequence Read archive. Okay. Thank you. And finally, just to show you this while I take any questions, as these are the links to both of the different resources I've shown you. Thank you.
Hi, so for this session, I'll talk about a resource that is known as NetMiner. So I guess earlier on in the questions, a lot of people are trying to connect some of these genes with their functions. So I personally find this as a very good tool. So NetMiner was built with the philosophy of building these genome scale knowledge that can interconnect data and enable effective knowledge mining and gene discovery. So information from other crops or model organism are being generated on a daily basis, and but they can often give you a clue as what this gene can be doing in wheat. But nowadays, a lot of data are being generated, and it's very difficult to follow up on them. And also sometimes you have people use different gene names depending on the model organism that they're working on. So it becomes very difficult to track them. So I feel like NetMiner is very good at connecting these information together. It creates a lot of public data sets, and then also search for key terms in the literatures to generate these knowledge network that can be easily visualized. So you can see the connection between your data set and your publication. And I often use NetMiner to help support generating hypotheses that can then be tested by experiments later on. So for this section, I'm going to go through 2 examples how I usually use NetMiners. to support my research. So the first example, white-grain wheat lacks flavonoids on the seed coats, but is also prone to pre-harvest sprouting. So historically, people has noticed a connection between the seed coat color and also pre-harvest sprouting as well, but they don't know how these two traits are connected. So we'll go to the example of using the wheat R gene, which controls the seed coat color in wheat, and we're going to try to find out how it can be connected to pre-harvest sprouting. So first thing we do is we usually type in the name of the gene here. Here you can see I use the 1.1 version 1.1 gene model. And right away, you can see that it has identified hundreds of connections with other publications or other work in other species. And honestly, it's very daunting if you are looking at it for the very first time. But I think one of the really powerful things that you should notice is that it can connect your gene of interest, in which case this is the R gene, with its ortholog in Arabidopsis, which is TT2. And right away, you can see a lot of work has been done in Arabidopsis already, right? It's been connected to let me see, flower color, protein content, or seed coat color. So there's a lot of connection there already. So you can imagine how this tool can be very powerful. So imagine you have a gene that you don't know its function. You can right away put it into NetMiner and be able to have a general idea what it does in Arabidopsis or in rice. And then because there's so much information, you can also filter it down with some of the key terms that we're looking at. So in this case, we're looking at seed coat colors connection with pre-harvest sprouting. So I put in two key terms, pre-harvest sprouting or seed dormancy, but you can add more. You can add germination and other stuff and so on. But right away with this filtering, you're reducing the connection to a handful that you can look at. And then if you just right click on it, you can show the info and see what are some of the evidence that are supporting this connection. For example, right now you're seeing a connection between our R gene and the NFT gene. So this is based on a some mapping results here that suggest MFT is involved in seed dormancy, right? And then, so you can make that connection. I always recommend reading, following up on that paper, just to make sure that you agree with the connection that they're making on this website. So based on the information we got, we can already come up with a very strong hypothesis that we can test via further experiments. For example, we can hypothesize that the R gene, which we know control wheat grain color, can potentially influence pre-harvest sprouting by regulating the expression level of MFT, which we also know is involved in regulating seed dormancy. So I remember there was a lot of questions earlier on QTL, GWAS, and stuff like that. I also think NetMiner is a great tool to kind of prioritize candidate gene after you do your genome-wide association study or QTL mapping. So usually you're left with the region that is associated with your trait of interest. And it can be hundreds of genes in this region. But with NetMiner, it can hopefully help you identify some of the most likely candidates in which you can design your experiments. So we're going to use an example that came out from the lab. So previously we're looking at these locusts that control gloom length. We're able to fine map it to 1.68 million base pair region on chromosome 6A. and we're going to use NetMiner to identify our candidate gene. So in this scenario, we put down our 5 map region. So for example, chromosome 6, your start position is here, your end position is there. And then right away you see 14 genes that are within this interval. So that's still a lot to look at. So then I just filter it out with my trail. It was gloom, right? So I just put gloom on there. So right away after the filtering process, we're left with 1 gene, which is SVP. So this is very exciting, and then we can then dig deeper into this connection. Right. So NetMiner was able to pull out some publication that is related expression of a gene also from this family in weeds also leads to an elongated gloom tissue as well. So then after going through so I hope after these 2 examples, you can appreciate how powerful it is, and then maybe use it to support your research in the future. So the next part of the presentation, Catherine and I will talk about a tool that can be used to verify the hypothesis that you have generated. So with NetMiner, you might have found the genes that you think control your trait of interest. How do you test it, right? And I think one of the best tools to go about it is to use the wheat tilling population. It's a very useful functional genomic tools, and it's still the dominant non-transgenic method for obtaining mutation in your gene of interest. So you can identify plants that knock out your gene of interest and then phenotype them afterward as a way to verify their function. So first, the seeds are treated with EMS, which is a mutagenic compound to produce mutation across the genome, and they're usually point mutation that makes the GC to AT transition. And these mutations can in turn affect the protein that the gene codes for. For example, you might have a missense mutation. So here it turns from one amino acid to the other one. Or you might have truncation, which can happen via a premature stop codon. So to find out what mutation these plants have, previous study have sequenced about 1,500 tetraploid weeds and 1,200 hexaploid weed via exome sequencing. So this means that they only sequence the gene coding region, which is really all we need for this purpose. And it's also so the sequencing information is then mapped back to the reference genome. So then we can rapidly identify which line out of these thousands have a mutation of interest. All right, so to read more about it, you can check out the corresponding publication. So as Nikolai already alluded to it, ensemble plants is very powerful, as usually where I do most of my analysis. So first you can search for your gene of interest. And then on the left-hand side, it's very small, you will see a section called genetic variation. So if you click on it, this is what you'll see in the tab. So you see all the tilling mutations that are across the gene of interest. So for example, we have a red one, which is a premature stop codon. So the mutation in this case was from TGG to TGA, which introduced a very early stop codon in our gene of interest. And by clicking on it, you'll also find out which of the plants carry that mutation of interest. In this case, it is cadenza 0113. So you can also view it in a table format if you select the variant table. So here you'll see a table of mutation with its corresponding plants that carry that mutation. So you can see the stop codon is very early on in the gene. So it's the 49th amino acids. This might be a useful mutant to follow through. However, the whole mutation process is random, right? So you don't always get a really good early stop codon. to work with. So you might be looking for some missense mutation that might affect gene function. So if you study a certain family or a certain group of genes, you might know certain residues are important, then you can look for lines that have mutation in that position. But more often than not, we don't have that information. And in this case, I would rely on the CIF score that they have provided. So this score is calculated based on the hypothesis that amino acid residues that conserve across evolutions are more likely to affect gene function when it's mutated. So CIF scores calculate how likely you are to see this amino acid in being changed in other species. So in this case, 0 means all the other proteins, all the orthologous proteins in other crop species carry the same amino acid, in this case glutamate. So very likely, if you mutate this amino acid, you might disrupt the function of the protein. So usually we look at, if the score is from 0 to 0.05, we predict it might have deleterious effects on the protein function, so it's labeled in red, while the green one probably doesn't affect the protein function. And of course, you can export the data just by clicking the export data tab as well. So some of the stuff that we consider when we're ordering when we're deciding which tilling mutant we want to order, of course, an early premature stop codon is great, but you don't want anything that have stop codon on the very end of the gene because it might not do much. Then you can also look at spike site mutation because some of these mutations can then also introduce early stop codon as well. So as we have touched on, it's difficult to predict the effects of missense mutation unless you have prior information. But in this case, you can look at the CIF score to help making that decision. So these are some of the more important part, but some of the other stuff that you can also consider is zygosity. So the lines are sequenced at M2 and some of the mutations are still heterozygous in the second generation. So typically you get the seeds from M4, which is later on. So if you select a heterozygous plant, you might need to do some screening in your M4, in which Catherine will touch on how to screen these mutants. So that's something to consider as well. And of course, it's also important to consider the population. If you're working with tetraploid plants, it's easier to get a complete knockout than in comparison to working with hexaploid plants. But if in your research program you're working with hexaploid plants anyways, then you might have to choose the Candescent population. So in short, I think tilling population is very powerful as a tool, but Catherine, how do we order these tilling mutants?
Once you've selected which mutations you want, how do we then order seeds of the plants that will contain that mutation? Well, you remember this little screenshot from Andy's part of the talk. We can click on this more about link or type this ID of the variant into the ensemble search box to get more detail about this specific. And this tells us the tilling line name. So here it's cadenza 113. And it also tells us that seeds can be ordered from the seed store. This website this seed store is maintained by the Germplasm Resources Unit here at John Innes Centre.
And we're going to visit it on Friday.
And they have a specific tilling cart where you can directly go to ordering your tilling line. So in this case, you just need to put cadenza 113 in that search and go ahead and order the seeds. For research purposes, they charge a nominal £15 per line. And then there are other agreements available for commercial purposes. If it are Chronos lines, then we have a mutation in our A copy of the gene and the B copy of the gene. We then need to cross them together to create a double mutant, because it's quite likely that those genes have overlapping or the same function, and therefore we need to knock out both of them in order to in the phenotype. We cross together our A and B mutants. This creates an F1 seed that will carry both mutations as heterozygous. We then solve that and get the whole range of possible genotypes, thing from wild types to single mutants to double mutants, and also a bunch of heterozygous lines, which are here. With the hexaploid cadenza, it's the same principle. The only difference is you then have 3 mutations to cross together, so it takes an additional generation. One thing to note is this timeline of 4 months per generation is kind of based on maybe growing these in a greenhouse, under standard greenhouse conditions. If we use speed breeding conditions involving 22 hours light, which is explained more in this paper, we can actually reduce this down to 3 months per generation, and therefore 9 months for tilling lines is that they contain background mutations. Line has a few thousand mapped mutations in different genes. At the phenotype we see is down to our gene and not any of these others. There are 3 ways to deal with this. One is to back cross the material, and this halves background mutations every time you do a back cross. So it could be quite useful for breeding application. The second is to compare against the segregating control line. What do I mean by this? Control that you want to use is this wild type that comes out of F2 population, because that means that while it is wild type at the mutation of your gene of interest, it still contains the same sort of background mutations as in your mutant line. So it's a fair comparison.
How do you identify this control ones, like do you use markers or?
Yes, I'm about to come on to that. And the third one is to select 2 independent tilling mutations. So kind of do 2 crossing programs for the same gene. And if you want more on mutations, this is the paper to read. You just asked, how do we track which one's a wild type and which one's a mutant? We can do this using Cas markers. Can I just have a bit of a hands up? Have you been told about Cas markers earlier this week? Okay, excellent. Well, you can use Cas markers for SNPs in tilling lines. And so we need to get our sequence-specific wild type primer, mutant primer, and sort of reverse common primer. And a useful way to do this in wheat is to use a website called Polymarker. This is specifically designed for making Cas markers for polyploid wheat. So here for our inputs, for each SNP, we need to input an ID for the SNP, which can be and which chromosome it's on, and thirdly, a flanking sequence. The SNP itself is highlighted with square brackets. Where do we get this information from? Again, Ensemble is our friend. We can take the ID from here and the chromosome, and then at an individual gene level, you can get the flanking sequence from this button, or the practical will show you how to get it from Bioinformatics. Output of Polymarker then gives us an A, a B, a common primer, which correspond to the wild type mutant and common primer I showed you in the first slide, and that's just the sequence specific sections of the primer. So you still need to add the found and hex sequences onto that. How does it do this? It basically, this tool behind the scenes is making a multiple alignment between the flanking sequence that you sent and the Chinese Spring reference genome. The SNP specific primer is highlighted in red here on the right, and the homolog specific common primer on the left. So it's found some base pairs, in this case, that are semi-specific, that is, they've distinguished the A and the B genomes against the D genome, but you're still going to get a little bit of contamination from A genome reads. Basically, fully homolog specific primers that target only the subgene of interest are, but if you can't get them, in some cases, you can get away with using semi-specific or non-specific primers. You can download the data here. Occasionally, Polymarker will not be able to design an optimal primer set, usually due to repeat regions or high GC or something like that. At that point, if you still really want a primer for this SNP, which you do if you're working with tilling lines, the best way forward is to try designing a few primer sets by hand. Some of you might notice this anyway, so I might as well tell you. There is a shortcut to getting Polymarker design primers for tilling lines on the variant page for some variants. But during the practical, please have a go at using Polymarker anyway. And if you go away from this and you come back and think, actually, what was it they said in this session this morning? There's a great website called wetraining.com that includes resources for tilling mutants, designing Cas primers and crossing schemes. And it does have some stuff relating to some of the other things we talked about this morning. Thank you.
You want copy?
Okay.
No, okay. I'll be back here and